In this video, I will be examining how the inversion of chromosomes can affect the viability of gametes. To demonstrate this, I will be looking at an example of a reproductive endocrinologist who is examining the chromosomes of two of her patients to determine if any abnormalities may be present that could impact their ability to reproduce and the potential need for alternative fertility therapies or treatments. These are the original chromosomes of patient 1 presented to the doctor. The letters represent different locations on the chromosome, and the period represents the centromere. The chromosomes can be better analyzed by flipping the bottom chromosome, which will allow the ends of the top and bottom to match. The flipping of the bottom chromosome will also show a flip of the middle portion of the chromosome. This creates an inversion. Because the inversion contains the centromere, this is considered a paracentric inversion. If the inversion did not contain the centromere, it would then be called a paracentric inversion. This inversion forces the chromosomes to create a loop, which will allow them to pair up during meiosis and allow for crossing over. This is how the inversion loop for patient 1 would appear. The normal chromosomes are represented by the top two lines, and the bottom two lines contain the actual inversion. So if a crossover took place between locations J and K on the chromosomes, the gametes presented under the loop would occur and are written in corresponding colors with their path on the loop. The gametes produced would be GHI period JKLM, GHKJ period ILM, GHI period JKHG, and then MLKJ period ILM. From these results, only the blue and yellow gametes are considered to be viable, whereas the red and green gametes are not viable because they are both missing a portion of genetic material, or an aberration occurs. So, the doctor will have to tell this patient that only 50% of her gametes are viable. On the other hand, the doctor's second patient doesn't have any aberrations on her chromosomes that would affect her fertility. Like we did with the first patient, we must flip the bottom chromosome. This causes them to be identical, and although the centromere was moved in this instant, it will not impact the viability of her fertility, as shown in the next slides. Since the chromosomes are identical, they can pair in a normal fashion, and there is no need for an inversion loop. The crossover would again occur between locations J and K, but it would ultimately still result in 4 viable gametes, as presented as GHKJ period ILM. So, the doctor would be able to tell her that there is no need to explore any fertility alternatives, as she would have 100% viability of her gametes.